Good evening. Am I audible? Good evening. Am I audible? All right. So today we will be talking about Unit 6. So, Unit 6, I will take up the first two units today, the first two part today, which is photosynthesis and plant respiration. Now, photosynthesis and plant respiration, they are very important from your CSIR exam, and a lot of questions are asked from these two chapters itself. In fact, plant respiration is one of that chapter which is similar to your animal respiration, because overall the plant respiration and the animal respiration is same except for two or three things here and there. Because in plant case also you will see there is glycolysis, there is Krebs cycle. In animal also you will find glycolysis, Krebs cycle, all of these. Okay. So and photosynthesis, as we know, this is a very very important process that happens in plants, which is sustaining the entire life on earth, isn't it? So these two topics I will discuss a few questions. Today, I will be discussing only five questions. And while I discuss the five questions, I will just, discuss the pathways also. What is happening? Why is it happening? Because I do understand that this is a process which is not very popular among the students, these two topics. So, shall we begin? Can you give me a bring it on BIO in the chat box? Okay. How many of you are from the botany background or you love to study plants? Let's see. Is there anyone who likes to study about plants? No? My. Okay. So let's start with today's question discussion. So the first question is saying Rubisco enzyme is involved in both reductive and oxidative carbon cycles in plants. Following are certain statements regarding them. Statement 1, sugar are produced in both the cycles. Statement B, pyridoxine is reduced only in oxidative carbon cycle. Statement C, product of oxidative cycle is one of the substrates of reductive cycle. And statement D, NADP and ATP are used in both the cycles. So they have given a comparison between your reductive and oxidative cycles. So C3 and C2 both. Right? And as we know that Rubisco enzyme, this is actually the full form of Rubisco is what? Ribulose 1,5-bisphosphate carboxylase oxygenase, right? So Rubisco has got dual function. It works both as your carboxylase as well as your oxygenase. Okay, though however we mostly use the carboxylase property, we exploit the carboxylase property in the C3 cycle, and that is what is desired. However, in many cases we do have the oxidative the oxygenase uh, version of it also being active, and that is the C2 plants. Okay, wow. Satyam is very fast in answering this. Option 4 is the correct answer. What about others? So, what about others? Okay, the correct answer here is option 4. So, basically, sugars are produced in C3 as well as C2 cycle. And in C3, what it is known as your reductive carbon cycle, right? So, in C3, sugar is produced from your glyceraldehyde 3 phosphate. Okay, from glyceraldehyde 3 phosphate, we get the sugar which is glucose. And in the case of C2 cycle, which is oxidative carbon cycle, this involves the conversion of 2 phosphoglycolate to 3 phosphoglycerate. Okay, which then further enters into the C3 or reductive pathway. So, if I look at the overall process, okay, first let us understand the overall process. So, in the first case, what is happening? There are three molecules of CO2, okay, which gets converted to six molecules of 3 PGA, phosphoglyceric acid, okay, or phosphoglycerate. And the first step is known as your carbon fixation step. This 3 PGA gets converted to six molecules of G3P. Okay, there is a two-step process here, but this two-step comprises of the reduction process where 6 ATP is utilized and 6 NADPH is utilized. Now, where are we getting this 6 ATP and 6 NADP? If you recollect, we get this from the light dependent process of photosynthesis, right? Because Kelvin cycles and your oxidative cycle, these are all your dark phase reactions of your photosynthesis, right? All these sugar producing cycles that you have. So ATP and NADPH, this energy units, we get it from your light dependent processes. So we get this six molecules of G3P, all right, and from here, five molecules of glyceraldehyde 3 phosphate is utilized to regenerate RUBP. So we get back three molecules of RUBP. This process is known as your regeneration. All right. And in this process, again, three ATPs are utilized. And one molecule of 3GP is used in the production of half a molecule of glucose. So basically, we need, so from three molecules of carbon dioxide fixed, we are just getting half molecule of glucose, right? So we need six molecules of CO2, right? Which is getting balanced here. Can you see? So we are getting six molecules of CO2 to get one molecule of glucose, right? Now, this is your C3 cycle or the Kelvin cycle. If you come to your C2 cycle, in C2 cycle, what will you see? You will see here that there are first and foremost three organelles which are in use: chloroplast, peroxisome, mitochondria. Okay, so here RUBP is fixing O2. This is why we call it as oxidative cycle. Here it is fixing CO2, but here it fixes O2, right? Now, because it fixes O2, you're getting two molecules of P glycolate, okay, and two PGA. Now, this glycolate, this is a useless product, and these needs to be converted. So, glycolate, to convert this glycolate, you need to undergo a lot of steps. There's a lot of conversions which is needed. That is the reason why again we say that C2 is not wanted in plants. Okay, this is a very expensive process, because see, in this process you are utilizing so much of energy in the conversion. So glycolate, that will be transported to peroxisome where it will get converted to glyoxylate, to glycine will be transported into mitochondria. From glycine you will get serine will be then again transported back to peroxisome. From there to it will form the hydroxypyruvate to glycerate will be transported to chloroplast, which is going to get converted to PGA, which is going to enter the C3 cycle. Ultimately, it is entering the C3 cycle only, right? And then again, it's you are getting this glucose, and you're regenerating back IUPP. Okay, so there's a whole lot of steps involved here. So, but in both the cases, we are do getting this sugar. So that is why the statement A is correct. Okay, sorry, statement A is incorrect. Okay, now since product of the C2 cycle, which is your 3-phosphoglycerate, as we have seen, this enters into the C3 cycle. It acts as a substrate for C3. Okay, there's a reductive process. So that is why statement C is correct. Okay, now 1 ATP is used in your oxidative pathway, but NADP is not utilized in C2 pathway. All right, so that's why D is also incorrect. Now, regarding statement, so A is wrong. Okay, A is wrong. B. Now, let's talk about B. See, this statement is saying that ferredoxin is reduced only in oxidative carbon cycle. Well, ferredoxin is definitely reduced, okay, in presence of light, and it further reduces the thioredoxin. Now, this thioredoxin system activates many enzymes in the C3 reductive pathway. Okay, now this thioredoxin cannot get reduced without FD getting reduced. Okay, so FD transfers the electron to your thioredoxin, and it reduces it. All right, so this is, yeah, you can say that it is a part of the oxidative carbon cycle, but if I be very precise, this is not directly involved. Indirectly it is involved. Okay, but if I have to choose from these options, well, I will always choose B and C. Got it? So this is how you have to analyze. Okay, all right, now moving over to the next question. So, question number 2 is saying the following statements are made regarding conversion of pyruvate to acetyl CoA from glycolysis to citric acid cycle. They have given you 5 statements. Oxidation of pyruvate to acetyl CoA is reversible. Pyruvate is transported into mitochondrion by a transporter. Pyruvate is carboxylated by pyruvate dehydrogenase. Acetyl lipoamide reacts with coenzyme A to form acetyl CoA. The flower proteins dihydro lipoyl dehydrogenase containing flavin adenine dinucleotide is involved in conversion of pyruvate to acetyl CoA. So you have to select which is the correct combination of statements. Okay, so what should be the correct answer here? Who will tell me which is the correct answer here? So if you analyze, you will see that pyruvate. Okay, first and foremost, before even I get into the explanation, this is a question which is asked from the chapter respiration. I hope you can identify that. Pyruvate, acetyl CoA, right? These are all related to respiration, not photosynthesis. Okay? So, if you see, pyruvate is oxidized to acetyl CoA and CO2. Okay? The correct answer here is option 4. So, pyruvate is converted, is oxidized to acetyl CoA and CO2. Okay? The overall reaction catalyzed by pyruvate dehydrogenase cluster complex, which is a cluster of enzymes, multiple copies of each of three enzymes located in the mitochondria of the eukaryotic cell, okay, and in the cytosol of the prokaryotes. So this is an oxidative decarboxylation. So therefore, B is correct. Okay. Now, oxidation of pyruvate to acetyl CoA and CO2 is an irreversible oxidation process. All right. And this process, in this process, carboxyl group is removed from your pyruvate, okay, as a molecule of your CO2. And the two remaining carbons becomes your acetyl CoA. So therefore, statement A and C is correct. Oxidation of pyruvate to acetyl CoA is irreversible, and pyruvate is carboxylated by pyruvate dehydrogenase. Sorry, A and C is incorrect because here they are saying it is reversible, which is wrong. It is irreversible, right? So this statement and this statement is wrong. Okay, now let's analyze further. So this pyruvate dehydrogenase complex, if you see, it is composed of multiple copies of three enzymes. Okay, it has your pyruvate dehydrogenase. Okay, with, and this has got its bound cofactor TPP. It has got the second enzyme E2. It is represented as E2, which is your dihydrolipoyl acetylase, transacetylase. Okay, and this comes with this covalently bound lipoyl group, and you have the third enzyme, which is dihydrolipoyl dehydrogenase. Okay, and this has a cofactor FAD and NAD. So this enzyme 1, this catalyzes the first decarboxylation of pyruvate, and in the process it produces hydroxyethyl PP. Okay, and then the oxidation of your hydroxyethyl group to the acetyl group happens. Okay, now the electrons from this oxidation that goes and reduces the disulfide of your lipoid bound, which is bound to your E2. Okay, and the acetyl group is then transferred to thioester linkage with your one of the acid groups of the reduced lipoid. So E2 catalyzes the transfer of the acetyl group to your A, coenzyme A, and as a result you get this acetyl CoA. So the E3 catalyzes the regeneration of the disulfide group from the lipoid electrons, and that obviously first passed to the FAD and then to NADH. Okay, so therefore statement D and E is correct. So overall, if you see, option 4, statement B, D and E is correct. Now coming to question number 3. Question number 3 is talking about the following are certain statements regarding PS2 electron carrier during the light reaction of photosynthesis. Statement 1 is saying the first electron released from reaction center P680 is transferred to QA to produce a plasto semiquina. Statement B, QA is a mobile plastoquinone. C, the first electron transferred from QA to QB converts QB into plasto semiquina. And D is saying QB is tightly bound to the complex and it is not mobile. So, which one of the following options represent the correct statements? So, the answer is option 3. Okay. Now, this is your electron transfer chain. So, if you see, you have two photosystems, photosystem 2 and photosystem 1. Okay, now this is what is happening in your light reaction of your photosynthesis. So PS2 you have, which is having the reaction center P680 and chlorophyll A P680, and reactions PS1 has chlorophyll A P700. Okay, so when light falls on PS2, all right, it excites this photosystem, and it releases an electron. Okay, not only that, even when your water is getting reduced, okay, it passes the electron here, so it gets excited. The electrons are then captured by your pheophytin. Okay, pheophytin passes the electron to your plastoquinone, which passes it to your cytochrome B6 complex, right? Then it passes it on to plastocyanin. From here, the electron enters photosystem 1. All right. Now, for photosystem 1, you have several electron acceptors here, and ultimately it is received by pyridoxine, which then transfers it to NADP, and this NADP gets reduced to your NADPH2. Okay, so accordingly over here, a QA is definitely not a mobile plastoquinone. Okay, and the first electron transfer from QA to QB converts QB into plastosemiquinone. This is these are wrong statements. So the correct option is only statement A, right? I hope this is clear to everyone. Is this clear to everyone? So this is also known as your non-cyclic electron transport. Okay, so you have QA, QB here. Okay, so these are known as your plastoquinones. Okay, so first it is received by pheophytin, and then QA, QB. So these are your plastoquinones. Okay, this is also known as your Z scheme. All right, let's move to the next question. It is saying pyruvate regenerated by glycolysis is converted to acetyl coenzyme A, which is metabolized by the citric acid cycle, regenerating energy rich molecules from the choices given below. Select the right combination of these molecules produced from one molecule of acetyl CoA. So, will you be able to, the answer is option 2. Okay, I've given you the option. But will you be able to do the stereometry yourself? So, they are saying combination of molecules produced from one molecule of acetyl CoA. Right? So for this, you have to know the entire cycle. You have to keep in mind, you have to memorize the citric acid cycle. Because from glycolysis, the pyruvate is entering the citric acid cycle. In the process, it is broken down to your acetyl CoA, and that is entering the citric acid cycle. Right? So, can someone tell me where NADH is used, where FADH 2 is used? Can you tell me? Just the steps. Let's see who can tell me this. Where these conversions are happening. Who remembers? Because this is important, you do get a lot of questions from the stereometry. So it can be asked from your citric acid also, it can be asked from your glycolysis also, it can be asked as an overall process of respiration, it can be asked from your photosynthesis as well. Okay, so wherever you have this NADH, FADH, the energy utilizations do go through the stereometry of it. So anyone? I've already given you the answer for this. The correct answer is option 2. So before I get into the explanation, I want you to try yourself. So when you study unit 6, particularly these two chapters, photosynthesis and respiration, please study in details. You do get a lot of questions from these two units. I mean, these two topics. Apart from the other topics of unit 6, these two topics you'll get a lot of questions. And it comes in different ways. Okay, and these two are the only uh, chapters which involves these cycles. So this is your citric acid cycle. So as you can see, acetyl CoA, it is entering the citric acid cycle, right? So citrate synthase is synthesizing citrate. Citrate is getting converted to isocitrate by acotinase enzyme. Here you utilize a molecule of water. Isocitrate is getting converted to alpha ketoglutarate, where you utilize a molecule of NAD, which gets converted to NADH. This happens by this enzyme isocitrate dehydrogenase. In the process, you produce a molecule of CO2. Then this alpha ketoglutarate, okay, from this isocitrate, you're getting alpha ketoglutarate, right? Alpha ketoglutarate is converted to succinyl coenzyme A by alpha ketoglutarate dehydrogenase. Here again, you are converting NADH to NAD. Sorry, NAD to NADH. Okay. Here again, a molecule of CO2 is formed. Now, from your succinyl coenzyme A, you are converting it to succinate, okay, by succinyl CoA synthetase. And here, GDP is converted to GTP. All right. From succinate, you form fumarate. In the process, the succinate converts to fumarate by succinic dehydrogenase. In the process, FAD is getting reduced to FADH2. From fumarate to malate is being formed by the enzyme fumarase. Okay, and a molecule of water is used here. Next, from malate, again you produce oxaloacetate by malate dehydrogenase. Okay, and here NADH is getting reduced to NAD is getting reduced to NADH. All right, again from oxaloacetate you form citrate. by citrate synthase using acetyl CoA. So, can you see how many molecules of NADH you are producing? One, two, three. Okay, so three NADH, one FADH2, and one GTP. I hope this is clear to all you. I hope this is clear to all of you. Okay, now moving on to question number 5. The last question. So they have given you a figure here. The figure below depicts the. Okay, for this one, I will remove this answer for the time being. Depicts the absorption spectra of chlorophylls and carotenoids over a range of wavelengths. Okay? And these peaks are given to you. So you have to identify which peak is corresponding to which one. So, see, this is the start of your photosynthesis chapter. When you start with photosynthesis chapter, you have to know the wavelengths of light, okay, the spectrum of light. You have to know the pigments which are associated with photosynthesis. That is your chlorophyll, okay, and you have the carotenoids. You have to understand what is your photosystem, okay, what is a photosystem, what it is comprised of. Antenna complex, red drop, immersion effect, and then obviously you have to understand the structure of your chloroplast, thylakoid, stroma, and then you have to understand what these where the process is, like the difference between the light dependent process and the light independent process. Okay, what is that, and what is happening, where is it happening, like whether it is happening on the stroma or whether it is happening on the thylakoid. These small small details you first have to understand, you first have to learn before you get into the chapter. If you straight away go into the chapter of Kelvin cycle and all, you will get thoroughly confused. And after you have studied these small small details, then you have to go into the light dependent process, the one that we did just before this question, the Z scheme. Okay, and you have to understand what are the molecules which are produced, where it is being utilized. So step by step, then you will come to C3 cycles. C3 cycle is utilizing the reductive property of Rubisco. Sorry, the carboxylase property of Rubisco. Okay, so from there you have to understand, we have seen that Rubisco also has the oxygenase property. Okay, which is when that is being utilized, then you are entering the C2 cycle. Okay, the plant is undergoing C2 cycle. Now obviously C2 cycle is not desired one, we have already seen that. So what will plant, so when does this C2 cycle happens? Okay, and then in when which conditions you get C2 cycles, and then And then you have to understand like which plants, what kind of modifications the plant has to to avoid the C2 cycle. So that will bring you to C3 cycle and camp. Now what is the difference between C3 cycle and camp? Okay, when you do this chapter in steps like this, okay, you will see that you have completed the entire chapter, you have understood the chapter, and you will start liking the chapter as well. Don't jump, it comes as a story, it flows as a story, one leading to the other. All right, so we will end the discussion with the topic which comes first in the chapter itself, that is the chlorophyll and all. So remember this. Okay, in every book you will get this picture. So chlorophyll A and chlorophyll B, they have two different peaks. They don't have the same peak. They have two different peaks. Okay. So and then you have chlorophyll B. Okay, and then you have carotenoids, which is around somewhere near the 500 nanometer you have. So you have the three peaks here. All right. So if you look at this picture, then you can see that the correct answer here is option 3. Okay. So okay, so A is corresponding to chlorophyll B, B is chlorophyll A, and C is representing carotenoid. Okay, peak C is representing carotenoids. So these are the different kinds of questions which comes from your photosynthesis as well as your respiration. Okay, so from respiration, okay, I told you about photosynthesis. Now let me give you a briefing about the respiration part. So respiration, obviously start with your glycolysis, then you have the Krebs cycle, and then you go for your electron transport pathway. Okay, these three things are important from your respiration part. Now, what's the basic difference between your plant respiration and animal respiration? Nothing much. In the case of animal, you have something known as breathing, right? Where the oxygen, by which the oxygen is getting in the plant, you don't have anything as called breathing. Now, where does this respiration occur, in which organelle? Mitochondria, right? Now, here again, you have to understand which cycle is taking place where. Like Krebs cycle is taking place where the glycolysis is taking place where electron transfer is happening where. Okay, see, these details you have to understand. You have to the sociometry is very very important, like how many ATPs, NADPH, and all these are produced. That is very very important from that part. And yeah, the enzymes. Okay, so if you study plant respiration, automatically you will be covering the animal also. So this is common to both your unit 7 and unit 6. Okay, respiration part. So my suggestion to you all would be that don't miss out on these two topics from unit 6. All right, Unit 6, all the topics are important. I'm telling you this repeatedly because you get a lot of questions from Unit 6. But if you're still selecting, if you have decided not to study Unit 6, I will tell you, don't leave out Unit 6 completely. At least do these two topics. All right. Of which, as I told you, one topic, respiration, is common to 7 and 6, and one extra you have to do is photosynthesis. Because the number of questions, if you take the weightage of Unit 6, and particularly the subparts of Unit 6, you give a lot of weightage to photosynthesis. All right. Apart from that, Unit 6, all the other important topics would be your nitrogen metabolism, it would be the stress, the hormones. Okay, hormones, biosynthesis of hormones, and also the functionality of the hormones. uh, then you have that osmosis, water potential, okay, transpiration, that part. So which is very easy, transpiration, nitrogen metabolism, these are very easy. Okay. uh, this hormone needs to be memorized, the functions of the hormones needs to be memorized. And you have the stress. Okay. So this is about your Unit 6. Okay. Okay. While we are discussing this, let me tell you one more thing. That is, I mentioned CAM and C4, right? So, and I told you what is the difference between CAM and C4. That also you need to understand. So always remember that CAM, that is crassulacean acid metabolism, and C4, both these two cycles, they are specially adopted, or they are not adapted to minimize the oxidative property of your carboxylase, because mostly the oxidative property of carboxylases are shown when there is very less carbon dioxide, that is in very dry and dry arid regions with high temperatures. Under those in those environments, you know, the plant can exhibit the oxidative oxygenase property of your rubisco. Okay. Now these C4 plants, the camp plants particularly, they grow in the dry areas. So they have made this adaptation. All right. What they have done is, in this case, they have not kept rubisco as the primary CO2 fixing enzyme. Okay, but rather they are utilizing uh, pep carboxylase. Okay, the first one, which is going to fix the CO2, and then they bring it to the C3 cycle. Okay, they concentrate the CO2 into the inner regions, and they give it to the C3 cycle where the rubisco is present. Actually, not C3, they give it to rubisco. So rubisco here, since it is now having a very high concentration of CO2 in the vicinity, obviously will be exhibiting the carboxylase property only. Okay. So in this case of your C4 cycle, you have the Kranz anatomy, where you will see there's a very distinct anatomical difference between C3 and the other plants. Okay, you have the bundle sheet cells, you have the mesophyll cell, you have the bundle sheet cells. So do go through the Kranz anatomy. And in the case of camp, the camp does the same thing. Okay, it also doesn't utilize rubisco as the primary enzyme, but rather it fixes CO2 and then, you know, transfers it to rubisco, so that rubisco only exhibit its carboxylase property. So in this In these plants, what happens is, basically the plants here, since they are growing, the camp plants, right? I'm talking about the camp plants. Since these plants are basically growing in the dry and arid regions, so in order to reduce transpiration, they keep their stomata closed during the day. Okay, and they open it at night. Now understand this. You have light only in the daytime. Okay. Not at night. So photosynthesis will be taking place during the day. But if you're opening the stomata at night, that means CO2 can enter the plant at night only, not during the day. So how does the plant balance these two things? That is very interesting to understand. So basically, if you see, during the night, the plant allows CO2 to enter and it fixes the CO2. Okay, keeps it in the vacuum. Fine. And then in the morning, when it is daytime, the plant, obviously the stomata is now closed, so no more CO2 enters, but CO2 has been fixed into a compound. Now this is released from the vacuole, and now there is light, so you have a lot of ATPs and NADPH which has been generated. So now you give this CO2, you break down the compound, you give the CO2 to Rubisco. Okay, and Rubisco, utilizing the energy units, it, converts the CO2, it fixes the CO2 once again to produce the glucose. Isn't it very interesting? Okay, so I'm not going into the details of it, the products and the things, everything. You read it yourself. Okay, you read it yourself. After, again, I'm giving you some time to read. You will be provided with a link of questions like how you get every time, and I will give it. The link will be given to you, but it will get activated after 15 minutes. Okay, so in the meantime, just go through these things. The question will be based on your photosynthesis and plant respiration. All right, is that okay? So, uh, go through it. You're going to find the things very interesting. Plant has its own way to deal with the problems. Okay, and if you want to know more about the plants, go into the details of it, then obviously you can join us. We will help you to clear all the ideas and you know discuss more questions with you all. So what do you feel? Will you be able to do the questions? What's your opinion? I've given you almost a summary of Unit 6. So, what is your opinion now? I hope you will be doing this unit now. Definitely do this. Okay. So, I will end my discussion here. Thank you, everyone.